Okay, hello everyone. Welcome to this video. We're going to feed back on some of the required practical work that you did last week. Thank you for getting all those plans in. Those of you that have not done so, please make sure it happens. Have a look through this. We're going to give some top tips on writing practical plans and look at some of the things that you've done well and some of the things you really need to work on. So, as a reminder, I asked you to plan an experiment looking into threats affecting the frog population of the world, or the amphibian population of the world. Right, so there's our lovely frog, right, who we're supposed to be helping. So we'll talk about the first things, the first obvious things. Right, there are some things in this practical that a lot of students did, which I really don't understand. Right, and there's a lot of this where certainly when we're asking for ecology planning and referencing tasks, there's an awful lot of stuff that people do that I really don't understand why you're doing them. So first things, these are blindingly obvious, should be a given, should be absolutely straight, done, no problem. If you're asked to fill in a sheet, you fill it in. The number of people leaving big blanks in big obvious boxes, it's the next one, big red obvious boxes saying fill this in, this is what we're looking for. The amount of people left them blank is bizarre. I continue not to understand what it is that is this massive blind spot that students have about referencing. All right, referencing should be dead easy. Okay, you put in the reference of where you found the information that you've just written about. Simple. Okay. If you're asked to submit in a certain format, please use that format. Okay. I've had everything from photographs of notes, PDFs of hand scrawled things like that, despite my request for no docx's so I can actually mark it and you can see the feedback. A lot of docx's coming through, which make life quite tricky. So if you're asked to submit in a certain format, use that format. Okay, and if the project is about amphibians, do something with amphibians. Okay, not birds or trees, both of which we had as submissions. Okay, completely different random projects. And of course, put your name on things. And this is not me just being fussy. This is a list that I actually checked and confirmed with a number of university lecturer friends of mine. And they say it is things that undergraduate students do. Right, they do all the same things, and if you do them at undergraduate level, so you don't submit in the right format, you don't fill in the right boxes, you don't do the task that's been asked, or you do something on a different subject, don't read the instructions, then it's immediate disqualification from that unit and you get a 0. Okay, so get that into your heads now that these are the basics you'll need to get marks at both A level and further study, degree level. Okay, these are all things crop up at university and they cost students dearly. Right, that's the tedious admin-y stuff that should be blindingly obvious out of the way. So let's get into the nitty-gritty. The questions. I think I made it very, very clear that everything really stems from a good question. Okay, if you've got a really good question, then you're going to have a really, really good experiment. So a good specific question makes everything else so much easier, and I could read through a lot of them where people were getting tangled in knots over the methodology or the stats. The reason was they didn't ask a specific question. They didn't know what they were testing, so therefore how can they make sure that there's a stat test or know what equipment they're supposed to be? So the basics, and again, this is GCSE level, you should all know this. How does the independent variable change the dependent variable? So the independent variable is the one I change, and the dependent variable is the one that you measure. What are you measuring? Okay, if these independent and dependent variables are measurable things, great. Okay, so for example, how does an increase, that's a specific directional change, in the mass, time, pH, number of something, these are all measurable quantities of x, whatever it is your independent variable is, affect the length, height, crispiness, flexibility, whatever, another measurable thing for the dependent variable of y. Okay, that gives you straight away, well, you know what equipment you're going to need, you know what you're going to need to measure, and it gives you a nice easy way to put the results table together and know what stats test you're going to use. If you don't have that, it's no wonder the rest is really difficult. Okay, so a bad one, how does x change y? what is X? How are you measuring it? What is Y? How are you measuring it? What do you mean by change? Do you mean go up? Do you mean go down? Do you mean both? Do you mean neither? Do you mean whether it exists or not? What are you talking about? And lots of the questions had very sort of humanities style questions in there. To what extent? Well, that's great for an essay. It's not great if you want to do some stats on it. Okay? You need a more specific question. Sure, statistics can tell you about degrees of things, but you need to be very specific about what you're asking. Okay? So, if your question was a bit awry at the beginning, and I think I made this very, very clear, it did make the whole rest of it a lot more difficult. Some of you managed it with difficult, unspecific questions, but those of you that did a nice specific question found the rest of it very, very easy. So I'm going to show you some examples now, and obviously no names required, of ones that I liked some things about and that I didn't like some things about. So there's all positives and negatives in all of these. So here's the first random student response I'm going to show you. What effect does pH have on Bacteridium dendrobatidis? I'm never any good at the Latin. The ability to cause chytromycosis in frogs. So what I like about that is it's got specific diseases in there, specific names. What effects does pH have? So that's a specific, measurable, independent variable. I can instantly see from that what measurement this person is going to take. They're going to measure the pH of something, and it's going to be on the pH scale from 1 to 14. Okay? Measurable, specific, nice. Perhaps what I don't like about that one is the what effect does on the ability to cause chytromycosis. What are we actually measuring? Is it the presence of chytromycosis on the frogs? Is it the level of keratin in their skin? Is it whether or not they've been killed? Is it mortality? Is it infection? What is it? A minor tweak to that question would be absolutely perfect. But that's a really strong question. There's a lot of good things about it, which will make some of the measurements, some of the later bits really easy. It's also quite tricky. This one, however, is really good theory, but this is a copy of the textbook. Okay? So it's not a question. Yeah? The distribution of species is determined by the range of different variables. These can be grouped into abiotic and biotic living factors. Abiotic factors include light intensity, amount of water, nutrients, and temperature. Biotic variables include competition for resources and number of predators and disease. Thanks, I think. It's not a question. And then the only bit that gets towards a question, whereabouts is the fungus affecting the frogs? What fungus? Whereabouts? What do we mean by that? Do we mean where on the frog? On the legs? On the head? Right? Do we mean where in the world? Do we mean in a pond? In a river? What are we talking about? In trees? What are we talking about there at all? Right? The fungus, which fungus? Affecting how? Killing them? Making them sad? What are we talking about? The frogs. Which frogs? Where? Okay? So that one, the theory is all true, but it's not a question. It's a copy of the textbook. Okay? This one is quite a good question, or could be quite a good question. And this one's had a couple of iterations, a couple of things changed about it since I first saw it, but it is not the topic that's being studied. I'm trying to find out whether one small section of forest is more biodiverse than another small section of forest, where one of those sections of forest contains a footpath, more human activity. So I can see what that person's driving at, and it's a valid question to ask. Does a footpath decrease biodiversity? But it's phrased in a very long-winded and peculiar way. Okay, so that one could be more efficient, and it could be about the right topic. It could be about frogs. Okay, so we could say what is the abundance of wildflowers in an area of forest with a footpath through it and not? Okay, that would be a valid question. Okay, more biodiverse. How are you measuring that? What are you going to measure? What is the actual variable you're looking for? From that question, it's quite hard to then go on. Okay, got another one here. To what extent has the impact of amphibian chytrid fungus affected the distribution of tree frogs in the Cusco National Park? Now, of course, that's the place that our video speaker was talking about in Honduras. They've taken that. It's a nice specific location. That person's obviously watched the optional video, knows that's an important place for frog conservation. So I like that. That's good. To what extent is an essay question, not a question that you're going to end up doing some stats on. Okay, so what we wanted here was a nice, clear, specific question. That one is a lovely essay title. but it's not really a question that you can test and design a methodology for. It probably leads to lots of different questions, which is fine, but we were looking for something specific you could research and you could reference. Okay? Right, so, and then this one, has the chytrid fungal pathogen reached Sri Lanka's amphibian population? That is a nice binary question. Yes or no? Do we find any chytrid in Sri Lanka? Easy, right? The dependent variable, right, is going to be, is there any chytrid here? Have we found any? The independent variable, right, is where we are, right? In Sri Lanka, not in Sri Lanka. Great, really nice and specific, simple, testable, good stuff. The only thing I would say is any. It's always risky in science to say any, because there might be like a, you know, an importer of exotic pets who has it in a shop, and they would never sell that. They were going to test that, swap that frog, and maybe never let that one out. It's in quarantine. Maybe there's a conservation organisation who are looking after frogs and curing them of chytrid in Sri Lanka. Therefore, there kind is some chytrid in Sri Lanka, but it's not in the wild. Therefore, it's not significant. You know, so that's the only criticism I would have maybe of that one. And then, what effect does the abundance of bacterial-generating dendrobatitis, terrible Latin, have on the populations of axolotl in the east coast of Mexico? Nice question. One is assuming we've got some method for looking at whether or not there is, when we say abundance, we mean, I don't know, number of cells on each frog, or number of frogs, number of axolotls with the disease. What are we talking about? But I can see how that would get to a nice, clear, independent variable. Have on the populations, so the population numbers. That's a nice specific count. And we've got a specific location in the east coast of Mexico. It might be good to say compared with somewhere else. Is there an axolotl hotspot? Or are we comparing it to numbers of axolotls 15, 20 years ago? Is this over space? Is this over time? I can see what the dependent variable here is. You know, the measurement of the disease. I can't really see what the independent variable is. So great dependent variable, not so much on the independent variable. And lastly, does the prevalence of chytrid fungal infections in frog species differ in Australia compared with Malaysia? Good, nice independent variable there. 2 countries, bosh bosh, here and here. That makes that independent variable nice and easy. Prevalence of chytrid fungal infections. Okay, so I'm thinking that person's leaning towards counting numbers of frogs or percentage of frogs that have the fungus. It says in frog species. Are we focusing on particular species? How are we going to compare like for like? Do Australia and Malaysia have the same frog species? Right, probably that could be explained a bit later on in the methodology. So good solid question with both things. So there's just a few examples. Let's move on to the methodology and the references for that and the equipment, the next few bits. Basic advice. This was something I sent this back to a lot of you about. References have actually got to link to something in the thing being referenced. So many times there was a methodology, an equipment list, something like that, and I clicked the reference below and it was nothing to do with methodology or equipment. Well, that's not a reference then. That's not a reference for that thing. You've just made something up and then put a link to something vaguely related to frogs. Not what you're being asked to do. The skill here is, can you reference your material? That is a required A-level skill. And there was also differences in the strengths of the references. Strong references, things like scientific articles, research reports, method sheets, all that kind of stuff of similar experiments that you could borrow from. Great. Weak ones, and I genuinely got all of these things. Wikipedia. I mean, it's a good starting point, Wikipedia, but Wikipedia is extensively referenced. You can look at the references in Wikipedia and you can take them further on and you can basically follow those up and actually get to where the actual research is. Wikipedia should be a starting point, not an end point. Okay? Genuinely got some Google Maps links. There was one talking about doing some studies in freshwater in some area near Henley and Arden, and one of the references was a Google Map link to head to the map of Henley and Arden. I believe you that Henley and Arden exists. You know, I know that. I don't need you to prove that. What I need you to prove is what's the method for. You know, that's the kind of thing. Magazine articles, Lovely background, didn't really have too much in them on methodology, on this is actually what a scientist did in the field. They were all, oh, isn't it terrible that the frogs are all dying? That doesn't back up your argument, doesn't back up your method. GCSE level or below material, sorry, it's not really recognised at A-level standard, right? You should be aiming higher than that. And stuff that's not referenced itself. So, yeah, not that I didn't see this very much, but quite a lot of people will reference things that is just made up. You know, if you found conspiracyjoe.com and they were saying, well, frogs don't exist really, they're just the larval stage of the royal family, right? Citation needed. Obviously, that's not a good reference, right? Oh, well, that guy on the internet says it, is never a good reference for anything. Okay, so the idea of a good reference and what you were getting these CPACs for and what would have got you sent it back if you didn't achieve this is the idea is to back up and justify what you are proposing. That's what you need to evidence and that's what we need to see. Okay, so let's look at a few examples here. So this one, to start off, this one is a weak method. Right, but we weren't looking at strength of methods in this particular experiment. We're looking at strength of references. Okay, so we've got make a random sample of frogs in ponds in 2 countries, one affected and one not. There's a lot of questions there. How? Make a random sample of frogs. How? What frogs? 2 ponds. How are you choosing the ponds? In 2 countries? Which countries? How are you deciding which is affected and which isn't? If there's just a little bit, are you saying that's fine? How much chytrid is affected and is not? And record whether the frogs have the fungus or not. Well, some of them might have the fungus, but showing no symptoms. Some might be dead. So you're going to draw no distinction between those? And then compare the populations of the affected frogs in one country, the population of affected frogs in the other. So you're just looking at the ones you deem to be affected, and you're going to compare them. How? What are you going to measure? Oh, there we are, look, there's 2 frogs, brilliant. Right, that one's green and that one's red. What are we talking about? Okay, so very weak method on this one, but look at the references. Okay, look at the references there. There are loads of good references that are, you know, A-level A-level sort of practicals over here we've got, A-level notes, fine. And then biology fieldwork.org, A-level fieldwork techniques. This does, all this stuff here, does reference very well a methodology and a equipment list, right? What it does do as well is show how lazy the person was in actually reading the methodology, because if they actually read all these references, they could have done a much better job of the method than that. But as far as what we're assessing goes, which is the referencing, that's really good. Okay, and the dates there that they were referenced, that they were accessed, perfect. This one, even worse, right? This is a terrible example. This is basically just nicked off the model answer that was up for a while. Literally word for word what was on Mrs. Omani's kind of example answer. Randomly sample a set of frogs in country A, an affected country. That's literally not an experiment, it's not a method. Okay, that's basically for us. That's a mark scheme for us to say, have they done a method that covers these things? And then as you can see there, that's an article. It doesn't have anything about methods in. Literally what the person's done there is gone, frogs, chytrid, first result, copy, paste, done. Okay? Doesn't back up an argument, not that an argument for a method is made. So that would be a hard fail. Okay? This one, much better on the method, but look at the gap. You can all see that, right? That it's big, red box. You've got to put some stuff in here. This is what we're assessing. Nothing in the box. Why? Okay, so we got a good solid attempt at a method here. Very like that very much. Okay, grid the habitat and use it. So, but there's a few things here which make me think the person's not really thought through their method. So we've got gridding the habitat and use a random number generator to pick coordinates for the transect to go through, which is representative of the different habitats within an area of a rainforest. So what we're using the random number generator for here? To pick coordinates for a transit. So we're doing like some kind of dot to dot. So we're going to have here, 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 and we're going to have the transect kind of going round them. I mean, it's a novel approach, fine. And then it says through which is representative of different habitats. If you're doing it randomly, how are you making sure it's representative? Okay, that's sort of a contradiction in terms, okay, within an area of rainforest. it might not be feasible to do this. And considering the distance that that this person's talking about, lay a belt transect, which kind of contradicts what was said at the top, in a straight line for 10 miles. So we're going to lay a belt, a wide strip, and we're going to lay that for 10 miles. That's not ecology, that's building a road. That's a different thing, okay? Through some non-deforested rainforest, commonly known as rainforest. Every 500 metres place a frame quadrat. Well, you've already said you've got a belt. Why are we placing a frame quadrat? What you're talking about here is an interrupted belt transect. So fair enough. We're going to walk 10 miles. It's a long quadrat, that's for sure. A long transect. It's possible, but it's a really long one to be feasible. Record the number of frogs. Yep, and the different species of frogs in the quadrat. Great. Record the number of peat swamp frogs per quadrat. Now, what would be really useful there is a reference to say why this person has picked particularly on peat swamp frogs. Why peat swamp frog? What's the point? What's that all about? A reference there to say these are particularly good indicators of chytrid, or they're affected by chytrid, or they're not affected by chytrid, or they should be abundant in this area and they're not, or something in the question would make it a lot clearer why we're suddenly going on about peat swamp frogs. Okay, repeat this 10 times. So we've walked 100 miles. Okay, I see an issue there. To get a representative sample, so we'll calculate a mean number of frogs per quadrat. Like that, that's good. Average number of frogs per quadrat, brilliant. Repeat for a 10-mile area in a deforested part of the rainforest in Borneo. Fine, great, we'll do that. That's good. We've got a comparison, nice, clear, independent variable. I like that bit, that's good. And calculate the mean number of peat swamp frogs in each area, calculating the mean number of different species of frog in each area, and calculate the mean number of peat swamp frogs per quadrat. So there's an awful lot of calculating there. It would be easier to know what the person was going for if they were really clear about what the question is, about what they're trying to achieve. Okay, and that will be backed up nicely if they had some references. So that one would have been sent back to that person to actually put some references in. So there's a couple of examples of what I'm talking about with our methods. So some good work, some not so much. Right, on to control variables. So that all that stuff goes for the equipment, and we'll skip over that bit. Control variables. Now this is basic. GCSE should know that this is Key Stage 3, really, maybe even Year 7. Cannot be, so your control variables can't be your independent variable and dependent variable. So you can't control and keep the same the thing that you're changing and the thing that you're measuring, right? There's one of each. This is basics. There's one of each. One control variable, one dependent variable. So you change one, you measure one. Everything else, as far as possible, you keep the same. Okay? And this might come back to the persistent confusion which you need to get out of your head, which is the difference between a control experiment and a control variable. A control experiment is one you do without the independent variable, right? Without the thing that you're changing, right? So for example, a placebo trial is a control experiment, the placebo group. A control variable is an aspect of the study that you keep the same. Yeah? Keep them separate. Right. Now, I know it's difficult, really quite tricky in ecology to do this because you're out in the real world. There's so much stuff that changes all the time that you don't have any agency over. You don't have any ability to control. So it's what makes ecology and environmental study so difficult is this methodology bit. It's why you need to be really careful about your referencing and about what you believe the scope of the thing you're going to do is. So you need to think of the things that will affect your independent and dependent variable significantly. Now highlight that word because there's loads of things that might do. The precise level of humidity. Oh, today is 70% humidity. Yesterday was 95. Oh, we can't go out today. Well, maybe that might make a bit of a difference, but is it going to make as much difference as, say, you going out at night versus day, or in the winter versus the summer? Right? These are much bigger effects. Okay, so you've got to think about what is going to affect your experiment significantly. Also, they have to be things that you can control. You can choose what time of day you go out, what season you go out, what sort of area you go into and sample. You can pick all these things. You can choose your equipment. You can choose all those kind of ideas. You can't control the weather, right? You can't control the even distribution of frogs in a pond, right? You can't control that. So don't say these things that you can't control. And also, we need some sort of indication of how important the control actually is. Is this the big one? Is this the massive one? If you've got nocturnal frogs only going out at night, that is a big control variable. That's a very important thing. If you were looking for nocturnal frogs in the middle of the day, well, obviously you're not going to find them. Okay, so time of day is really important. If you're only going to go out when it's just rained, well, maybe that might have a bit of an effect, because frogs obviously lose moisture through the skin, that kind of thing. But it's not a massive control variable, and really your time in the field is probably going to be more of a limiting factor. So something to consider there. It's not just what is a control variable, what's important and what's not. Okay. And you might be sit there and ask the question, well, how am I supposed to know if the phase of the moon affects frogs as it does or something? And how am I supposed to know if, this? Well, that's what references are for. That's what this whole exercise is about. You're supposed to research, find people who've looked into this before, and put a reference in there saying, oh, I think phase of the moon is really important for these frogs because they only mate in a full moon. Here's where I found the information, right? That is research, that is referencing, and that is the skill that we're testing. Okay, if we can be absolutely clear about that. Right, so some examples of this, some good ones and some bad ones. Okay, so we've got here, look, control variables. So we've got the number of infected frogs from each country in Scotland and Wales, so one close to home. That is the dependent variable. Okay, that's fair enough. Take the count from an equal number of ponds in each area, like it. Use ponds with an equal or similar size. Might be practical, we'll have to define what equal or similar is in this context, but I like that thought that we're going to survey the same kind of area. We're not going to compare your garden pond to Lake Windermere. That's a good control variable. Use ponds that are unlikely to be disturbed by traffic or people, should be at least, so that's a good suggestion. And then even better, we've put a measurable limit on that. Should be 2 kilometres away from any road or house. Brilliant, right? I'm not sure where in the UK you can find a pond that is over 2 kilometres from somebody's house. Maybe you'll find enough for your study, but that's a really good specific control variable. Like that one, well done. Take the count of frogs at the same time of day for all ponds. Yep, very important, as we've just discussed. Take the count of frogs on days with similar weather. I like the fact if this is going down in the list of importance, good, because that one, again, the practicalities might be get in the way there. You might only have a certain amount of time to take this, and you might have to be a bit more flexible with it than that. And how similar is similar? What if it's the same amount of raininess, but a different amount of sun? It's hard to pin that down. And then this one here, the water quality lab here, that reference is good for some background information and does indeed have a little bit about methodology in it, but I would like to see a couple more references in there with a little bit more in terms of things that are being kept the same, because the amount that was pertinent to the control variables of that reference was very, very small. Okay, and in fact that reference, that article, actually had further references, if you followed those along, that went more to methodology papers that if you click through, you could have gone and found those ones. Okay, so this one here, look, use frequency count. Yep, fine, that's the dependent variable. Test, so this is a bit strange now, it gets a bit weird from here. Test frogs at a lab to see if they have a fungal disease. May not need to test them, as you could tell just by looking at them. Red skin and slower reflexes. There's a number of issues with testing frogs. How are you going to test that? And the methodology for this experiment didn't go into that. Collect them and continue to count in other areas. Don't release yet, as you could count the same one twice. Great, good. That's absolutely true. Good control variable. So you collect them all in, count, and then you release them at the end of your study period. Fine. Obviously, as long as that's not going to harm the frogs and you can keep them there, you might do some sort of mark, release, recapture on them, where you mark them and then you re-release them. Right? That might be so you know if you catch the same ones, and that can be useful. Control variable, same method of counting. Yep, fine. Frequency, percentage cover, same level of disturbance. So, e.g., be quiet so the frogs don't get scared. You're hoiking them out of their pond. I think they're probably going to be a little bit upset, but such is life. Same equipment. What equipment? Same random number generator to reduce bias. Is that a significant one? I know there's debate about random number generators and whether they're truly random, but is that a significant effect on what's actually going to come out of your experiment? I don't think so. The reason I chose to put this one up is because we've got two contrasting references here. Right, that first reference, really strong. It's got a really good methodology in it, lots of control variables, and it's a proper piece of primary research. Great. Then there's just bunged in there for good measure the Wikipedia page on chytriomycosis, the fungal disease. Okay, not exactly great. Right, it doesn't really tell us much about control variables or anything like that. So that one has two conflicting ones: a very good, strong reference and a very weak one. Okay, this one here. Independent variable location of site with varying salt concentrations. Yeah, good. Salt versus freshwater. Measuring with a conductivity meter, like that. Nice and specific. Well done. Dependent variable, the prevalence measured using LFD antigen tests. Fancy lab work. Like that idea an awful lot. That's very good. Control variables, species of amphibian. Fine. Geographical location, Eastern Australia. Now, that needed to be a little bit clearer, because location is also the independent variable, so it on its own cannot be a control. And there's also some far more. So if we're saying we're keeping it generally to Eastern Australia, but then we're looking at salt and freshwater, well, then there's other things we need to consider. Flow rate, things flowing through. Is the water moving? Is it not? What's the other local flora and fauna like? How are we going to control for different ecologies in salt water and freshwater? Okay, and also amphibians don't tend to live very much in properly salt water, so the different salinities could be quite difficult on a spectrum to get. But excellent reference, really good example, primary research, link to where it is and the date accessed, authors there, perfect work. That's textbook citation work, very good. So last thing then is the stats. Okay, they come from the question. If you didn't do a good question at the beginning, it's no wonder you would have struggled to come up with the stats. Okay, stat tests only test what we ask them to. They are literally a mathematical method of comparing two numbers or sets of numbers. That is all they do. They are not magical genies that you ask for great wisdom and it comes down. You really need to be specific about what it is that you're putting into them. Okay, so a clear question will give you a really clear choice of stat test. Okay, if your question clearly shows you're going to average something, then, well, you know that comparing averages gives you a t-test. If you're going to count the frequencies of frogs in ponds in one area versus another, well, you've got frequencies of things in categories. Therefore, it is, it's a chi-squared. If you're looking at salinity and you're going to have a certain frog in a certain salinity, or a certain amount of chytrid on a frog in a certain salinity, well, that's paired data, and you can rank that, and that's Spearman's rank. So a clear question leads to clear stats. Okay? And I want to introduce you to this little acronym that I like very much, NINO, N-I-N-O, which basically means nonsense in, nonsense out. Doing the stats test is really, really easy. Bunging numbers in and getting numbers out, your calculator will do that for you, right? The trick is picking the stats and asking the right question. That's what you're going to be assessed on in the A-level. That's where the skill is. Just bunging numbers in and getting them out is easy. Okay? So you need to be able to think, okay, what is the question that I'm asking? I don't think I can say that enough because it clearly hasn't gone in for some of you. So I'll give you some examples here. This person was on exactly the right lines. Really good job linking the question, which is at the top, to the stats, but kind of bailed out in terms of confidence in their answer here. So what's the difference in the abundance of amphibians along two transect lines in the Amazon? One belt transect runs either side of the footpath, whereas the other is 50 metres away into the forest. Great. I can see the independent variable, I can see the dependent variable. It's difference in abundance, meaning we're just counting stuff. So data is hard to do a statistical test of reliability. Is it? I can see a couple of ways you could do that. The only thing I could do is find the mean of two transect quadrats and work out an average. Brilliant. This is great. You know, do that. That's a really useful way to do. And then from averages, we do a t-test. Fantastic. You know, that's exactly a thing to do. Have some confidence in that answer. So that person should have been a little bit more confident in their statistical ability. However, that last phrase, the null hypothesis, is that the mean of both transect quadrats is equal? No. Right? You let yourself down at the end. There's no statistically significant difference. They're never going to be exactly the same, but is the difference significant? Are we at least 95% confident that the difference is or isn't significant? That's what a null hypothesis looks at. Okay? This person here, bang on. What effect does the velocity of water current have on the abundance of chytromycosis of the mimicking rain frog populations? We're going to use a chi-squared because we've got frequencies of things in categories. Used to compare the observed frequency table and the expected frequency table. Great. The only thing I would question there, which hopefully is in the methodology somewhere, is what's the expected? I mean, do we have prior knowledge of how frogs live in flowing water? No idea. Okay. What is the expected there? So that's the only question I would have. And then it says requirements, all expected values above 5. If not, must combine groups. I think I know what you're getting at there, and I think there's perhaps a little bit of an introduction of some bias, of some problems in that. But I wouldn't necessarily do that if that means what I think it means. And then the null hypothesis is no correlation between river velocity and the abundance. Yeah, absolutely right. I'd have a question about how we measure the abundance, but yeah, that's exactly what the null hypothesis should be. So nice work there. And also nice work here. Bit of a vague question, but kind of pulled it out of the bag with the method and then the stats. What's the effect of chytrid on the variation and distribution of native toads in the UK? Quite a broad, general question, but you're asking to do a t-test there, okay? The data. The mean number of toads with chytrid per lake in each region, fine. Normally distributed data, yes it is. Enough samples to calculate a reliable mean, yep, great. The null hypothesis could be there's no significant difference between the mean number of toads with chytrid for each region, and there's no significant difference between the mean number of toads of each species with chytrid. So which one of those you'd use depends on which question you're actually asking, but that is a brilliant suggestion for the stats tests, and it's all come from a good question. Okay, so that's really good. Now, a little recap on stats. You've only got 3 tests that you can choose from at A-level, right? There's only 3. There's more in existence, of course, and most statisticians kind of invent their own to suit the purposes. Chi-squared, which is frequencies of things in categories. Spearman's rank, which is the pairs of data that can be ranked. And student's t-test, which are comparing things that can be averaged. We are going to do the next lesson this week is going to be a statistics recap that the whole biology department are going to help us out with. You've probably had this diagram before, which is the little decision tree AQA put out to tell you what you should be using and when. It is important that you know this because exactly those kind of questions, what test you're going to use and why, are going to come up on every single statistical test question. Okay, you'll never be asked to do the maths. You're never going to be asked to calculate a stats test. You will be asked to know which one to use. So we're going to do some practice in the next lesson because it looks like we need it, but generally some pretty good job and pretty good work, and if you have corrections to do, please get them back to me ASAP. See you next time.